First, I want to thank Kevin so much for doing a lot of the legwork and organizing this conference. It was a tremendous amount of work, and I appreciate that. I also want to thank Hannes, our discussant, for jumping in at the last minute, the last hour, to discuss this paper, and we're looking forward to his comments. So this paper was very much a team collaboration, and all of my co-authors are in the room. So that's rare when that happens. So I want to acknowledge them, and they should also feel free to jump in if they have other people have questions. And so, yeah, let's get started. All right, so typical outline, we're going to talk about motivation. Then we're going to talk about the lung health study RCT that we used in this study, data measures, empirical strategy, results, discussion. And then if we have time at the end, I have one slide on some future directions that I find particularly interesting in gene by environment interaction research, and that is the use of variance polygenic scores and functionally annotated polygenic scores. So the motivation for doing this research is that, we and a lot of other people in this room think that genetic data can improve our understanding of heterogeneous responses to treatments or policy interventions, both because it's an important source of human diversity, and it's determined upstream from all of other outcomes. But also from a cost perspective, it's a source of heterogeneity that will be increasingly inexpensive to exploit in field experiments or with survey data. As we know, genomic data are getting cheaper and cheaper. So for the cost of about $40 for a typical array chip, you can investigate hundreds of PGIs without having to resurvey individuals. So this is something that we think might be increasingly used in RCTs in the future. However, currently PGIs seem to be rather underutilized in randomized interventions. RCTs are increasingly collecting genomic data. However, they're not yet utilizing PGIs. And I think this is really because genomic data are typically used in the context of precision medicine or pharmacogenomics. So these clinical RCTs are using genomic data to examine response to medications at the gene or SNP levels. They're doing, really looking at drug-SNP interactions in the context of precision medicine. But we think they could also have some interesting application for RCTs in economics and the social sciences. That using PGIs in particular may shine light on some potentially interesting variation in treatment effects, and could perhaps provide mechanistic insights at the behavioral or biological level. And in addition, it might help redirect or improve treatment outcomes for high-risk individuals, which might further reduce genetic disadvantage as the result of a trial. So there's been a lot of prior G/E research on health behaviors and economics by many of the people in this room. Some of my earlier work on the effects of the Vietnam draft lottery on smoking behavior, where we showed that high PGI individuals were more likely to become lifetime smokers as a result of being drafted into Vietnam. Work showing that high PGI individuals that have higher risk for smoking behavior are less likely to respond to higher cigarette taxes. Work on compulsory schooling reform by Sylvia and colleagues on showing that basically education can reduce the genetic disadvantage for high-risk BMI individuals. Alcohol licensing policies and in the UK and health inequalities, that work by Pietro, showing that people with a high genetic risk for alcohol use disorder are more likely to select into areas where they have easier access to alcohol and are less responsive to stricter licensing policies. And then work by Jason showing that the minimum drinking age, once high genetic risk individuals for alcohol use disorder turn 21, they're more likely to become binge drinkers. So all of these studies are showing that there's differential response to these policy environments by underlying polygenic indices. But as far as we could find, there's very few PGI studies to date that have used an RCT. We did find one interesting paper by Daniel Dick and colleagues where they were showing that family intervention, a randomized controlled trial in youth that was targeting, it was a family intervention to kind of help the family better communicate with youth that were identified as high risk, potentially behaviorally. And they did see that for these youth that had high PGI for alcohol use disorders, because of this intervention in adolescence, they were less likely to become dependent on alcohol about 15 years after that intervention. So some interesting evidence there as well. So based on this, we wanted to kind of use the lung health study RCT, which I'll talk about just a little bit as a potential case study for really looking into how perhaps we could look at interesting heterogeneity by PGIs in the context of a randomized control trial. So in this paper, we asked did the effect of a smoking intervention on smoking behavior, BMI, and lung function vary by underlying genetic predisposition. And again, we're utilizing data from the lung health study, which was a large multi-center RCT. So a little bit more about the lung health study. It evaluated the effect of a smoking cessation intervention on cessation maintenance and lung function. It was conducted between 1989 and 1994, and it was considered a successful intervention. So at about 5 years, 22% of participants who were in the treatment arm had sustained quitting. So they had quit cigarette smoking for 5 years, compared with just 5% of usual care participants. And I would say the original kind of objectives of the lung health study can be boiled down into 2 points. One was to determine if a smoking intervention and the use of an inhaled bronchodilator, so basically almost like an asthma inhaler, could slow the rate of decline in forced expiratory volume in 1 second, or FEV1, in smokers that were showing early signs of chronic obstructive pulmonary disease, or COPD. So FEV1, if you're not familiar with it, it's this measure of lung function. So you take a deep breath and then you exhale as much as you can in 1 second. The more you can exhale in 1 second, the healthier your lungs are. So it's a kind of a measure of lung function. And then they also wanted to, in addition, estimate the effects of bronchodilator therapy on lung function decline. At the time, there was some evidence that this could have additional therapeutic effects to slow the rate of lung function decline in these smokers. They enrolled 5,880 smokers who were aged 35 to 60 years with spirometric signs, again, of early COPD. These were heavy smokers. So these were smokers that were smoking around 30 cigarettes per day on average. This was done in 10 hospitals across the US and Canada. Each hospital recruited around 600 participants, with about 400 assigned to the treatment group and 200 to the control group. And the data includes 5 annual follow-up visits. So a little bit more about the intervention specifically. So the first treatment was a smoking intervention, which was an intensive 12-session smoking cessation program that combined behavior modification, the use of nicotine gum, and a 5-year maintenance program to minimize relapse. So basically at the beginning of the trial, if people were randomized into the smoking intervention, they were first seen by a doctor who told them, hey, you really have to stop smoking. These are the risks. You're already lung function decline. You're in the early stages of COPD. And then they were put into these behavioral sessions that were led by a health educator. They had 12 sessions over 10 weeks. So initially there was a lot over the first 10 weeks. And then after that, they continued to kind of help them if they needed additional cessation, behavioral modification programs, and so forth over the next 5 years. And then for some, they also received a bronchodilator in addition. So this was ipratropium bromide, or Atrovent. It was prescribed 3 times daily, 2 puffs per time, from a metered dose inhaler to decrease airway hyperreactivity and improve mild airflow obstruction. So participants were randomly distributed into 3 arms. The first was a smoking intervention plus placebo. We'll call that the I group. So these people received the smoking intervention, but then they just received an inhaler with no medication, so placebo inhaler. Then there was the IB group, we'll call them. They were the smoking intervention plus bronchodilator group, the ipratropium bromide. And then the usual care group, which will just, no intervention. There were several exclusion criteria. I won't spend too much time on this slide, but these had to be relatively healthy, heavy smokers. So no serious illness in the past 2 years. They couldn't be pregnant. They couldn't be binge drinkers, so consuming over 25 alcoholic drinks per week. They couldn't currently be using a bronchodilator. They couldn't have an allergy to atropine or bromine, which was what was in the inhaler. They couldn't have high blood pressure. They had to be likely to stay close to the treatment facility, so unlikely to move. And they had to be willing to enter this behavior modification program. So these were also smokers who were motivated to quit. In terms of prior literature on the lung health study, there's been a lot. So initially, the results of the trial showed that the treatment increased the likelihood of smoking cessation and did reduce the age-related decline in FEV1. There was no difference that was found in alcohol use at 1 year between the treatment and control groups. Seemed to result in lower rates of respiratory illness. A later study found that mortality was significantly lower in the treatment group. Studies in economics have found that quitting led to an average increase in BMI of about 11 to 12 pounds, 1.8 to 1.9 units. And then Jason has some interesting work showing that the spouses of individuals in the treatment group were also more likely to quit smoking. So some interesting spillover effects. However, as far as we know, prior studies did not find any differential impacts between the two treatment arms. So in other words, the use of the bronchodilator didn't appear to have any significant impact on lung function or smoking cessation. So these findings appear to be mostly driven by the smoking cessation, behavioral intervention. So data and measures, we're going to utilize information from all 5 visits. And the dependent variables we'll be looking at are smoking cessation. This was validated by cocaine measures or carbon monoxide levels, cigarettes per day, BMI, which was measured by medical staff, and then what we'll call the F ratio, so FEV1 over FVC. Again, this is max FEV1, it's the total amount you can exhale in 1 second over the total amount that you can exhale, period. So in that kind of standardization is necessary because people who have different sizes of lungs and so forth can exhale more air. So it's just kind of helping to standardize that. And then we used genotype data that was eventually collected for about 73% of participants. And it was collected in a subset of European ancestry participants for whom lung function data was available for at least 3 time points. As far as we could tell, there was no evidence of genotype sample selection, so we didn't see any evidence that people selected into, or that people who were treated were more likely to be genotyped, et cetera. And we also didn't see any observable selection bias between the genotyped and non-genotyped samples. So the PGIs that we constructed used imputed genotype data and weights from the largest GWAS that were available for the phenotype to date. So for smoking initiation, cessation, and CPD, we used the Liu et al. G-scan GWAS. For BMI, the Yango et al. GWAS, and then F ratio was a Shrine et al. GWAS. The site's down there if you're interested. And in terms of interpretation for some of these, and I'll talk about the smoking PGIs in particular, because that's where we find some interesting results. These are genetic effects that have been shown to be really strongly associated with nicotine metabolism, as well as nicotine receptor genes and pathways. So in terms of smoking intensity, which is what the CPD score is capturing, how much can you smoke? That's really, tagging genes that are related to nicotine metabolism, so how fast your body metabolizes nicotine. The smoking initiation PGI, on the other hand, is tagging these dopaminergic systems that affect reward processing and addiction. So it's really saying something about genetic predisposition to smoking addiction, to becoming addicted to smoking. And we constructed these using PRS-CS Auto that Hugh mentioned earlier today. That's utilizing a Bayesian framework to place continuous shrinkage priors on SNP effect sizes. So again, this is just helping to increase PGI prediction by readjusting those SNP beta weights from a GWAS to account for LD patterns. We run just a simple linear interaction regression. So here, Y is the outcome for individual I at visit T. Again, we look at quit status or sensation, so linear probability models for those, and then CPD, BMI, and the F ratio. Treated as an indicator for whether individual I was assigned into one of the treatment arms. PGI, we use the PGI for each outcome. So if we're looking at BMI, we use the BMI, PGS, et cetera, PGI. And we standardize these to have mean 0, standard deviation 1. And then we include a matrix of covariates, including age fixed effects, gender, principal components 1 through 20 of the genetic data matrix to calculate for population stratification. We also include visit fixed effects, and we use robust standard errors clustered at the individual level. And then what we did is we estimated these regressions separately for each treatment arm and for both treatment arms combined. So treatment definitions, again, IB is equal to 1 if participant received the smoking intervention and the bronchodilator, and 0 if the participant was in the usual care or control group. I was equal to 1 if the participant received the smoking intervention and 0 if they were in the UC group, and then the composite if the participant was in either the I or the IB group, and 0 otherwise. So first, this is just, we just kind of are recalculating what has been shown in the literature already, which is just the change in these outcome variables over time across visits by treatment status. So here you can see, this doesn't show, but here you can see just the change in foot status across waves. So originally everybody was a smoker, and then once the trial started, you can see that those who were treated in the composite group in either treatment arm, you see about quit status of about 40%. This is the cross wave quit status. So if you look at sustained quitting, it's closer to 22% across all waves compared to the UC group. Cigarettes smoked per day fell dramatically from an average of around 30 to about 10 in the treatment group. A slight increase in BMI, which is to be expected. That smoking is an appetite depressant. So after individuals quit, they tend to gain weight. And then what the literature showed as well, which was just a kind of slower decline in lung function in those in the treatment group. So their F ratio is better. So here you can see, their lung function still declined over time, but not as much as those in the control group. All right, so results. So first I'll show you the results for smoking cessation. And here we're using the smoking initiation PGI. So higher values of the smoking initiation PGI mean we're more likely to become addicted to smoking. We also do have results with smoking cessation PGI, which I won't show. I can if you're interested, but those were null in terms of the interaction effect. We think is in part because this is a pretty underpowered PGI. It's constructed in a much smaller sample of smokers. But we did find some interesting results with the smoking initiation PGI. So here you see on average in the treated group, individuals were about, if you were treated, you were about 23 percentage points more likely to quit. And we find that, however, if you had a high initiation or a high smoking initiation PGI, for every 1 standard deviation increase in that PGI, you were about 2.5 percentage points less likely to quit. So, high PGI individuals had a harder time quitting than those low PGI individuals. And it seems like this is in part being driven by the bronchodilator group, which is kind of interesting. For CED, we find a similar pattern. So treated individuals, on average were smoked 9 cigarettes fewer. Those in the high PGI group, however, we see an interesting effect, which is that they smoke about 1 cigarette less in addition for each 1 standard deviation increase in the PGI. So here you can see is the control group, higher PGI, smoking more per day on average. But in the treatment group here, we see that kind of just genetic disadvantage or that genetic gradient was flattened somewhat by the treatment. So we no longer see any significant differences between high PGI and low PGI individuals in terms of their for BMI, again, we find on average that the treatment increased BMI by about 0.4 units. And then we found an additional effect where low PGI individuals were actually more likely to gain a little bit more weight. We didn't really find as much difference here at the high end of PGI. No significant difference there between the treated and controls. But for individuals in the low PGI group, we saw a little bit more weight gain. We think perhaps maybe this is because they were already quite thin or underweight to begin with. The sample did have a fat tail. So perhaps that's what's going on, is when they quit smoking, making it a little bit more weight. We didn't see any, again, the treatment improved lung function overall, but we didn't see any interesting, G/E effects going on here. In terms of other analysis and robustness checks, we did look at the SNP level. So we did do a deeper dive to see if we could find any interaction effects at the SNP level. We didn't see anything. We think we were likely underpowered to perhaps do that. The results were robust to the inclusion of additional controls like marital status, college education, and also their interaction with the treatment. The results were robust to the inclusion of other PGIs and their interaction with the treatment, as well as the interaction of the PCs with the treatment. So we checked that. And then we also looked at these G/E interaction results over time. So we did find that, the results were pretty consistent for smoking cessation across all 5 waves. For CPD, the results declined slightly over time. And for BMI, those interaction results actually slightly increased over time. So just to wrap up, we find that treated individuals were 23 percentage points more likely to quit smoking, but that success really varied by their underlying PGI. So every 1 standard deviation increase in the smoking initiation PGI reduced your likelihood of quitting by an additional 3.5 percentage points. This treatment appears to have significantly reduced the genetic disadvantage in smoking intensity. So average CPD in the treatment group fell by about 9 cigarettes, but for individuals in the high genetic risk category, CPD fell by an additional 1 cigarette for each 1 standard deviation increase in the CPD score. For BMI, again, we found an overall increase by about 0.4 units and a kind of extra weight gain from each 1 standard deviation decrease in the score of about 0.3 BMI units. And, in terms of the magnitude of these effects, how should we think about this? We did look at other interactions with the treatment and things like marital status, having a college education, being female. And, the treatment effects are pretty comparable to those or slightly smaller. So in the case of marital status, that was a 7.8 percentage point increase in the likelihood of quitting smoking if you were married. College education, we didn't find a really strong interaction, interestingly enough. So that was pretty comparable to the genetic effect that we found. So having some college education increased the likelihood of cessating by about 4.6 percentage points. We didn't find anything here for CBD. We did find a strong female effect for CBD. So being female increases the amount that you smoke on average by 3 cigarettes. That's been found a lot in the literature. Women on average have a harder time quitting smoking, which is really interesting due to some nicotine interactions with different hormones and things like that. So not really a comparable effect there, but close in terms of CBD. In addition, as I mentioned at the beginning, prior LHS research did not identify any differences between the 2 treatment groups. And here we did see some interesting results where it looked like some of our results were being driven by participants who received this bronchodilator. And this is really hand wavy, so I don't want to say that this is what's going on, but we did speak with a clinician because we were interested if there were some potential additional pharmacogenomic effects that perhaps weren't detected initially, and as a result of this bronchodilator. And they did think it was kind of interesting because ipratropium is known as an antimuscarinic. So when you're taking this inhaler, it's kind of blunting these receptor genes that are involved in proteins that produce mucus in the lungs. So it's helping to decrease mucus accumulation in the lungs. And interestingly, it can also act on these muscarinic receptors in the brain. And it's been shown, at least in cocaine users, that this ipratropium can actually help decrease cravings in patients that are trying to recover from cocaine use. However, it's been less well studied in smokers. And what's interesting too is we find this in particular with the smoking initiation PGI. And that's a PGI that again is really enriched in the central nervous system for functions that are related to reward processing addiction. So it kind of makes sense from the standpoint that it might be decreasing breathing intensity. Or perhaps the bronchodilator had additional therapeutic effects. So it could have just decreased mucus production and airway reactivity, which may have incentivized participants to smoke less. But overall, more research is needed here. This is, just again, kind of a hypothesis. So just to wrap up, I think this is just a case study or an example of how genomic data could improve our understanding of heterogeneous treatment effects or responsiveness to interventions. I think for economists using PGIs and RCTs, it might also help improve our understanding of underlying mechanisms. So for about $40, you can analyze multiple dimensions of genetic risk or underlying biology that may be driving treatment effects. So in this case, perhaps if you found an interaction between the depression PGI and the smoking intervention on smoking outcomes, maybe there's some indicator that the behavioral modification is also working through some genetic channels that are related to depression, which is also highly related to smoking. That's just an example I'm throwing out there, but just to kind of get an idea of how we could use PGIs to kind of help us perhaps dig a little bit deeper. And in addition, I think that it also kind of might help towards high-risk genotype individuals who might need additional therapies or interventions to improve their treatment response. And then just in the few minutes that I have left, I think there's some exciting future directions for G/E work in the future. The first is this concept of a VPGI, or variance polygenic index. So a standard PGI is estimated from a GWAS of mean effects. But a question is whether this is really the ideal measure for capturing variance in an outcome that's resulting from differences in the environment. There's some new methods out there, one by Q's lab, Jiaqing and Q, and also by Alexander Young and colleagues, where they run what's known as a VGWAS. So here they're using methods that decouple the SNP mean and variance effects to estimate effects on phenotypic variance in the population independent of the mean. And what is believed to be captured here is what's known as variance quantitative loci, or VQTLs. And these VQTLs are effects that are changing or are motivating the plasticity of a trait. So how fast an individual responds to their environment. And these SNPs are different from other SNPs that might be tagged in a mean effect GWAS. So using a VGWAS, you can then calculate a variance PGI or PGI that's capturing aggregate genetic effects on phenotypic variability. So perhaps something to look at more in future studies. I do have one study down here where I found where I was looking at job loss and genetic risk for BMI, and I did find that the VPGI did a better job explaining the G/E effect than a standard MPGI or PGI. Another thing that I'm excited about, and Q talked a little bit about this today too, is again, when we look at a standard PGI, it's capturing the average effect of the SNP on a phenotype across all cellular states and tissue types. And that's not something we often think of as economists. We're thinking a lot about the environment, but there's also a lot going on biologically when we're just running a GWAS. So here you can see with a nerve cell, it's expressing certain genes differently than a muscle cell, but they all have the same DNA in their nucleus. So we're working with the Liu lab of Q again and Jiaqing to construct PGI that are cell or tissue type specific using functional annotation data. So for BMI PGS, you could construct over 200 different types of PGI to show how the BMI PGS is expressed in the liver versus adipose tissue versus the central nervous system or the brain. So I think this is a really interesting future direction, in particular to help perhaps identify biological mechanisms or pathways that are interacting with the particular environment that you're looking at. So I want to say thank you to our awesome postdoc Shuba, who's also on the job market, who did a great job with the data and helping us bring this project together. Jason, of course, Q, and Jiaqing. And yeah, that's all I have.
Yeah, thank you very much. I'm Hannes Schwann from Northwestern University. I'm very new to genetic research, and so I'm a health economist, generally interested in the interaction of health and human capital. And I guess my comments today will be more like from a, let's say, like an applied micro layperson perspective. And so I might be a little bit ignorant here and there, a little bit not very fluent in some of the terms, but maybe there is some general takeaways that could be useful. So just as a broad motivation of this paper, like of the bigger picture idea of this paper, is the RCTs are of course the gold standard typically considered of causality, but then we know they're often a black box with respect to mechanisms. Well, the one thing that we can do with RCTs without messing around with causality is to do a subgroup analysis. And that could be useful on the one hand to predict who is impacted. That of course can be important for policy. And then ideally we can even get a little bit more towards mechanisms to understand why effects are stronger in one group than the other. And so what the authors suggest is that we should use more genetic information to make those subgroup analyses. And I think that's particularly promising for two reasons. On the one hand, these are of course very objective measures. They are continuous, so you can potentially create a lot of objectively chosen subgroups that might just give you a lot of insights into who are the people who are most at least impacted. And then of course, in terms of causality, there could be potentially be a first order causal driver, in particular for outcomes, for health outcomes, or for, let's say, kind of like behavioral outcomes that are very closely linked to biology, as for example, addiction and those kind of things. And the authors also add that genetic measures are increasingly collected, relatively cheap add-ons, given how expensive many RCTs are. I think there's also, there's of course like an additional ethical issues. So RCTs are always ethical issues, at least to people who like me were exposed to a lot of Angus Deaton's paradigms. But of course, I always think like with the RCT, a good benchmark is like, could you run the RCT on a group of Harvard MBAs, and now think about just like asking at the end, like, oh, by the way, we'll also take your genetic profile. So there could be an issue about like what kind of populations would agree or would not agree to this additional information. The application here is a smoking cessation RCT. And the lung health study, and as an health economist, I was particularly interested in that aspect as well. It was quite an interesting study. So the one thing that I want to point out, this is a group of people who have chronic respiratory disease issues already, and they smoke a lot, 30 cigarettes per day. So from a European perspective, I'm from Germany and I studied in Spain. That's kind of like not so unusual. As from a US perspective, this is kind of like suicidal behavior, right? And it turns out the US perspective is right, because you see the treatment effect here on cessation is like 16 percentage points, and 15-year mortality declines by 15%, right? If we think about that, this is kind of like the complier group, right? Do the divide by the first stage. This shows that actually all the mortality came from not quitting, right? And like you also see that people like predominantly died of like lung-related stuff. So this stuff literally like saved people's life for those who comply. So it's really very, very impressive and like a life-saving RCT. Genotype data was collected for 73%. And so now what the authors do is that they interact the treatment with a PGI that is like specific to the different outcomes from GWAS, and get kind of like, I would say, somewhat mixed bag of results. On the one hand, smoking for the smoking initiation PGI that reduced the cessation effect. On the cigarettes per day PGI, that actually increased a little bit the treatment, that increased the reduction, but only for one for the treatment arm combined, these sessions with the additional like breathing, lung breathing medication. And then for the BMI PGI, that reduced the effect on BMI, and for lung functioning PGI, there was like no effect. And these were the type of figures that we have already seen. So And just, again, like a little bit of a layperson comments, just like what would be my applied micro perspective on those results. So I think overall, the idea is to get a better understanding what's going on at the RCT level, right? To get to this black box. And I think at that level of, at least what I've seen in the slides, it is remaining a little bit of a black box. And particular for me, as a layperson, but I also, I don't know whether you guys would have a direct answer to this question, like who are the people with the high and low PGIs, right? That's really what we want to learn a little bit more about. And I think in general, things that we can, that would be useful to show is, show the PGI distribution within the sample in terms of the population-wide standard deviations. Like what are the type of scores or indices that we see there? And then show descriptive statistics for the top and bottom index quartiles to learn who these people are, and then show the treatment and control effect, or like the treatment effects in a non-parametric way, just like plot really by the index quartiles. And finally show like how much do we actually gain in comparison to other covariates in terms of forming new subgroups, right? And I mean, this is a lot of tables, a lot of stuff, right? But at the same time, I mean, like, if you want to learn more, right, maybe, a little bit less is more in a sense, right? And before we actually understand what's going on at the bottom of it. And then I'm like always, I'm not a big fan of like plotting linear interaction terms, in this kind of predicted way, because I mean nowadays we are really used to having studies where like all of these are entered and we are used to data-rich studies where where each of those confidence intervals would represent an individual treatment effect, right, or like an individual like a data point essentially. Well, this is really just like a prediction. And the other thing is, I think those kind of like plots of linear effects can overstate a little bit the predictive range. So, for example, the study excluded obese, overweight individuals, which is 25 plus. So I don't know exactly how that prediction like lines up with that, but it just sometimes it's like we don't even know whether we have anyone in that case. And again, like this is totally. I mean, this is standard, and I think it's just like more something like maybe a general takeaway. Then in terms of interpretation, what do you make of the results? I think it's important, to adjust for multiple hypothesis testing, because right, you're just like, having RCT, and we throw in like more subgroups and test more stuff. And also in terms of like the pre-analysis hypothesis, it would be interesting to discuss what is actually our prior, right? So we could think about, for example, if you have a genetic predisposition to, let's say, smoking, right? Then of course it might be more difficult to get away from smoking. But if we have two people who smoke the same, right, then we have to ask, why is the other person smoking? And if, for example, it's the social influences, and they might be much more difficult to change than you just your personal thing. If let's say all your family smokes, your friends smokes, right? In Europe, everybody smokes, and when I came here, I stopped smoking. So, this can be a very strong factor, and so it's not really clear in which direction it goes, I think. And then, of course, also when you talk about differences or treatment arms, you can check whether it's significant. When it comes to causality, this is always my question, and I want to talk more about you guys maybe in the break. And I think like the GUSs, that's right, that's just the correlation, right? But you have the perfect causal design when you look at sibling comparisons, right? Like as far as I understand, right? Like if you look at within family variation in sibling variation in genes, you have the causal effect of the genes there. And of course, they're not as largely available, but I think like in Danish data, you can do that in more data sets. You can try to get to the to actual the causal indices in a way. And I wonder whether this could be, if we want to get to causality, whether this would be useful to focus on maybe smaller studies, but that are more causal. Then one important question, I guess, is like, to the extent that we have, let's say, causal genetic variation, is it just running through the baseline characteristics? Just like people smoke more with certain genes, and that's why it's easier or less easy to have them quit smoking or reduce smoking. So I think like controlling for the outcomes, the baseline outcomes would be important. And then I guess again, like my lay perspective a little bit, just like always keep in mind that like the behavioral impacts of genes, even if you have like perfectly causal genetic factors, and then entirely a social construct, right? I mean, like again, like US, Europe is different. It's probably not because of genetic differences, and of course, the social mediation differs strongly across groups, right? And I think John mentioned yesterday, like some, the issue about representation in genetic studies. So this one is 95% sample wide. And I think one aspect there is not only to acknowledge that that's a problem, but maybe also talk a little bit about like what that means, right? That could really mean that in certain subgroups, effects might be entirely different because the social environment is so different, right? So it's not only saying like, oh, sorry guys, you're excluded, but we just assume that things are the same. It's like, no, maybe things will be extremely different, right? And so just like layperson's bottom line, when adding genetic data to RCTs, it's useful to be transparent and what kind of subgroups will form, especially, of course, in the study, typically, I think the authors control for a lot of observable characteristics, but that doesn't necessarily help so much because then you just know even less what kind of groups these are, right? It's just like, what is the residual variation that you're using there? And make sure it's not just picking up sampling variation, more causal genetic associations, the better, and then being aware of the social mediation. Then I think, on the one hand, from the outer perspective, like the genetic data is like so like, hard sciences and the something like really at the frontier of research. At the same time, you have this huge part of subjective interpretation of the researcher, and that is the social mediation that could really be most of the effect. Thank you.
Thank you. Yeah, John.
Well, I really like the paper and discussion, and I think it's very cool that, interacting the RCTs with the genetic, I guess it's like going way more sophisticated than what Mendel did, but I just wanted to, just point out that I came upon this study that was looking at use of naltrexone at medical. I don't know if you've seen the study. It was done at the Medical College of South Carolina, and they randomized people into treatment of naltrexone or placebo, and then they found the genetic interactions between a gene that governs opioid receptors and one that does dopamine. And they found these really interesting interactions that if you have high opamine, receptivity, which makes you more addictive, if your dopamine levels are low, you're more responsive to naltrexone. But if you don't have that interaction, it has no effect. And so they found some really cool interactions by using the RCT and look, but so the only comment there is they don't have many observations. They had 152 people. So I guess they have to invoke strong priors. They knew what genes to look at. And I just wanted to know if that's an issue for what you're doing as well.
Yeah, definitely. Because I think we didn't. So when we're using the PGIs, it is this kind of, we're thinking that the genetic pathways or the treatment might be interacting through a specific, spore, and we're thinking we're better powered, if we use a PGI. But yeah, most of these precision medicine trials, they're looking at these specific genes that they know respond to naltrexone. Naltrexone, I think, works on opioid receptors to blunt the opioid receptors. So in that case, they knew to look at those opioid receptors, and they might not have had to have been as well powered. In this case, we don't know. There's so many genes that are involved in like the smoking initiation PGI that we just weren't sure. Jiaqing did do a whole SNP search, but we just didn't find anything at the SNP level. So I think we were maybe just underpowered in that context.
This article did mention, though, there's some aspect of the addictive nature of nicotine that is related to the opioid. They didn't find a super strong interaction, but yeah, it's
Interesting.
Could I just follow quickly on this? But you said in one of your slides, if I understood it right, in these regressions that you controlled for the other PGIs, if I understood it correctly, and that your results were robust to that. But I guess I also wonder in this process of when you don't know for sure what dimensions, right, you want to look at, so you're trying different PGIs, unlike the study that John was just talking about. So I guess could you comment more on both those, what you meant by robustness there, and whether or not any of these other ones were interesting? And do you find anything?
Yeah, in that case, I think in that particular one, I would say that was more exploratory rather than a robustness check. But we were just interested, because these PGIs are also correlated to an extent, whether or not they, if include those interactions with those other PGIs, whether we saw anything interesting. And, you know, I don't think we saw too much there. I don't know, Shuba, if you remember more specifics.
But that's another issue of power.
Yeah,
Yeah.
potentially, yeah. In this case, I think the treatment seemed to really strongly be working through smoking in particular, so we just didn't see a lot of other, ramifications there.
Did you look, sorry, super interesting. I was just curious, did you look at any of like the mental health just because, you know, like smoking,
We did.
it's a way to
We did.
deal with stress and stuff, and nothing.
I was very curious about that also because I've done
Yeah.
some research on smoking in women and how that's really correlated with the depression PGI.
Right.
And there's some interesting biological interactions going on there. But yeah, we didn't see anything with any of the mental health PGIs. Yeah. So.
One question on that, is, and I think Hans had this in a point in the slide, so maybe a little bit repetitive with this, but I mean, I think, you know, in just thinking of what value added there is in bringing genetic data into an analysis of treatment effect heterogeneity like this, the first thing that I might, you know, try to do is say, well, could you replicate that type of interaction by looking at the baseline characteristics that are correlated with the PGI for initiation? Like, so for example, if that's, I don't know what that's correlated with, like the cigarette per day for these people, like at baseline.
Yeah, we did. Yeah, I didn't show that, but I do have a table that shows that the prediction value of the PGIs is comparable for the CPD score. So here was the issue with that, is that for the CPD score and the BMI score and so forth, we could check the prediction at baseline which, you know, was before the intervention happened. For the quit score, we couldn't do that in the smoking initiation PGI because at baseline, they were all smokers. So there was no variation there that we could look at how well it predicted.
Well, I mean,
But we did see comparable prediction rates to the
well,
G-scan.
What I'm saying is that would you be able to pick out groups of people based on observables that would sort them into the high sensitivity groups, right? The group that's being picked out by a high level of the initiation PGI, right? Are there observables that will let you sort of identify them? So, you know, so you think what would be a value add to this? I could go and target treatment, for example. Could I do that targeting on the basis of observables? In which case, the value added, it's not so clear, right? That maybe we should be running. We could have just been running a big analysis trying to find heterogeneous treatment effects based on observables. But if this is really going above and beyond what we can get at baseline observables, then that's really powerful. That's really powerful to say, yeah, we're learning something and our ability to target goes beyond.
Yeah, no, that's a really good point. Maybe I could show that slide, but we also did look at just the incremental R squared of things like sex, education, and I forget what else at baseline. Marital status. We looked at age, but that's a little.
I don't understand why that is informative of this. I mean, this is really a statement about whether R squared of the PGI.
No, incremental. So we looked at the incremental R squared of the PGI, but also of these other observables that you're saying. So at baseline, we looked at the incremental R squared in terms of how well, for example, marital status predicted quit status and how well.
It's not clear to me that that's informative about whether those observables would help you identify groups that are.
I guess I'm not sure what your question is or what you're asking.
You have lots of ways of predicting treatment effect heterogeneity.
Yeah.
Like, is PGI more powerful than other predictors?
Well, as we showed, at least in the ones that we could look at, so we looked at treatment effect heterogeneity by marital status, by education, and by sex, and it was comparable to those. Marital status was slightly higher.
Yeah, I would have, but if I'm from a clinical perspective, I would have stratified based on intensity of smoking or based on baseline flow volume. Like, I would really try to pick out the health variables, behavioral, manifestations of this that might lead you to believe that you might really benefit from this. I mean, you seem to be trying to get at kind of like baseline characteristics that are not endogenous to smoking and those outcomes, but like those are precisely the things that a clinician would.
I see.
try
So
to
looking
use
more
to
at,
target
I see. So you're saying, yeah, we could definitely look into that more. Yeah, it's a great.
Well, on this point, two things. First of all, when you introduce those additional covariates, do you interact them with treatment? And do you interact them with treatment and PGI?
Yes.
Okay, so and that would be interesting to get an idea of the magnitude of the PGI vis-a-vis, for instance, I would suspect age makes a difference. That it might be easier to stop when you're younger than older. I don't know, but there is no reason to. And the second thing is coming back to Kevin's point. one thing that's nice with PGI is that this is really exogenous. You know, it's something that people are born about because if you stratify based on observables like the quantity, you're stratifying on something that's endogenous. I mean, you're introducing all kind of selection bias when you do that, right?
That's fine, but if what I'm trying to use this for is to direct a scarce treatment in the direction of a group that will get the most out of it, it's not clear to me that I care whether I'm using, you know, information from endogenous variables or not.
Yeah, yeah,
Right. I mean, I guess it depends on the purpose. If
yeah,
what I'm
exactly.
trying to
Depends on
do is
what.
get a causal effect and then interpret that in some way.
Yeah, exactly. If you want an interpretation, it makes a difference. If you, I mean, from the
Do is use the intervention
Yeah.
to target the treatment. Then it's not clear that maybe I think
So it seems like one simple thing you could do is you could take all the baseline characteristics you have from the RCT and you could use some of these machine learning approaches to try and search for variation in treatment effects. So like causal force, for example, Susan Athey, but there have been more updated methods. And then you can try and sort of ask the question, there are ways of sort of diagnosing variable importance in these methods. So if you're using something like causal force, you get a measure of variable importance, which is basically like the weighted splits used in a variety of trees in order to establish these causal trees. And you can see how important is this PGI relative to other variables. But it does seem like the first thing that I would do, which was already mentioned in the comments, is to just look at like by quartile and look at the characteristics that you might think of as a first order being important, and seeing like, is there just obvious correlation with those things?
So back to your question, I wonder how expensive it is to just calculate this PGI for patients. I mean, make it a part of the treatment. I don't know. I'm curious, and I'm in general curious how expensive it is to collect the genetic data that we're using. Or maybe it's even possible to make this a part of your treatment,
23
collecting like the most important SNPs on them and use them as a proxy for figuring out who needs more treatment.
Well, yeah, it's the real cost of like $40. It'll be billed to insurance at $8,000, right?
40?
I'm joking. 40.
So it's possible.
So following up on what you said, I think it's what Kevin said. I think it's potentially useful to have genetic data and use it in randomized control trial, especially if you do something like a SNP by environment interaction and then use PGen or something like that, because it helps build a group of like a whole bunch of information that we had on like basically a lot of GWASs could be seen as taking all of the phenotypes that we care about and projecting it on the same space, so that different phenotypes from different cohorts from different data sets talk to the same, can be compared with each other through genetic correlations. And if we do that also with our CTs, then we create this type of information and we can see, look, this type of intervention was effective for those type of people who have these genetic predictions. So you start including in the same database, basically, information about responses to different treatments, and it can be systematically correlated with the genetic predisposition to phenotypes and so on and so forth. You're kind of building one by one a huge database that everyone can have access to. So I think there's a value in that, above and beyond whether maybe the polygenic index wins the rat race of the machine learning of what estimates more the heterogeneity treatment effects.
Yeah, I like that, yeah.
I just want to build on Kevin's suggestion of thinking about this in the real world context of allocating a scarce treatment.
Yeah.
Have you thought about doing a cost benefit calculation for like, this is an expensive intervention, clearly. Like, what are the gains in terms of cost effectiveness if you use the PGI to help select who's going to get the treatment? What are the gains relative to using what clinicians would already potentially be using?
Yeah.
And what are the incremental gains to adding the PGI to some set of standard
variables.
Yeah, no, that would be a nice addition, I think. Yeah, we've talked about it, so that's something, yeah, we could definitely.
Whether Sylvia, and then that'll be the last
Yeah,
comment.
I mean, just to follow up from Pietro's, I think the other beautiful thing about collecting genetic data with RCTs is that it's one of the best balance tests that there are out there, right? You know, you can even collect it after
Yes.
the intervention, and it's to predetermine. It's, you know, it doesn't change over time with interventions, and it's continuous, right?
Yeah,
So it doesn't get any better for a balance test.
Yeah,